Let's start recording. So if you have questions about the unknown project, you do need to get started on it. So ask me if you are having trouble getting started or if you're having problems, and I will do my best to get you started. If you don't start soon, you will not have enough time to get it done. But if you get started now, you should have enough time to get this project done. Sometimes, I'm not sure why, students wait until the end to work on it, and then they don't have time to get it done. So get started now on that project, and then you'll have time to get it done. Any questions about any of that? I should have mentioned in the lab that I did do some grading over the weekend, and I can't for the life of me remember what I graded, except I am grading one of the labs, which it's not finished, and I was hoping to get it done before class. And now I'm hoping to get it done after class because I never got it done before class. But I got some other things graded too, and I just don't remember what they were. So please take a look at what's been graded. Hopefully I'll finish. It's either lab 6 or 7 I'm working on now. I'm calling that information up, so just give me a minute. Come on. Yeah, I'm in the middle of grading lab 7. I got about half or maybe even over half done. Let's see about lab 6. I don't remember if I graded this one or not, so I'm looking at it. Now, this one you're supposed to have done by October 31st, so I didn't do lab 6. And I did grade at least one of the worksheets for the lecture. Maybe I got a quiz graded. Moving to the quizzes. Quiz 2A is graded. All right, any questions about that? Remember for the lab that when I get it graded, and hopefully I'll finish that tonight, you will have until Saturday to make your corrections. And you can get 100% as long as you get it in by Saturday. And that's 11:59 p.m. If you do make corrections after that point, you will need to email them to me and the assignment will be counted as late. And you will get partial credit for late, depending on how late you are, but you won't get full credit if it's late. Any questions on anything else? All right. I noticed that students are logging off very quickly after the lab, meaning after I start talking in the lab, and that's fine. You don't need me to work on the lab. However, I suspect a large number of you are getting together and forming your study groups then. And I'm going to request that you not work on the lab material in the study group. Obviously, I'm not going to stop you because I can't stop you. But if your group does do that, it would be better if you did not, and just be honest about that you got together in a group and you left that group because they weren't doing what they were supposed to be doing and you wanted to study or something like that. It's only 1 point of extra credit. And I'm asking you not to work on the current lab assignment for the extra credit. You're supposed to be doing things that you're studying for the quiz or for the final. And you're not supposed to be working on your worksheet for the lab. You're supposed to be doing that in the lab time. The extra credit is extra credit for doing something extra. And I'm trying to encourage you guys to study together and get the extra credit. I'm not trying to encourage you guys to get together and work on the lab. I have noticed that a lot of the lab answers are very similar. So I know that a lot of you are working together, and that's not a problem. You can do that. However, make sure your answers are your own. Do not just quote your study buddy, because if I do catch that, and I'm not really looking for it, but if I do catch it, you will both get a 0 on that assignment. Any questions about any of that? All right, meaning I want you to work together. That's not the problem. And you can share answers, just don't plagiarize, okay? Make sure everything is in your own words. And if ever you have to use your study buddy's words, you should put it in quotes and then say who you got it from. That is not plagiarizing. Any questions about any of that? All right. So today we're working on Lab 8, identification of gram-positive organisms. You were also supposed to be working on your unknown project and finishing up your infectious disease paper. Your infectious disease paper is due this Saturday, November 6th at 11:59 p.m. One student just emailed me asking for a topic. And of course, that was a topic that somebody else had taken. So I replied, they have to find another topic. And I told them what topics were taken. And hopefully they'll get back to me with another topic. Any questions about any of that? All right. For the lab, I'm here to help you. But once the last student logs out, I'm going to log out as well. Any questions about that? And like I said, lately that's been very, very soon, which is fine. I don't mind leaving early if you guys don't stick around. But if you have questions, I'm here for you. But once the last student logs out, I'm going to log out. Lab 8. I think this is the teacher's one. Yep. So identification of gram-positive organisms, we're going to be talking about gram-positive cocci, especially the Staphylococcus family, the Streptococcus family, Enterococcus, and then gram-positive Clostridium, Bacillus, Corynebacterium, and Listeria. There are some subtle supplemental materials, video clips, dichotomous key. There's some case studies. These last two are provided as links in Canvas. Are you guys seeing this? Are you seeing my screen?
No, we're not seeing your screen.
All right. Somebody has to remind me. I was talking like I'm doing it. I got it recorded. That's good. When I'm showing you my syllabus, the schedule, I mean to be showing you that. And then obviously the lab. The only time I'm not supposed to be showing you is when I have the grade book open. And it's true, I opened the grade book to try and remember what I had graded. But after that, I could have opened it so you guys could see it. And I forgot to do that. My apologies. Anyways, the last two, the gram-positive dichotomous key and the case studies, are links provided in Canvas. Let me show you that now. Anyone remember what week we're in?
7.
7, thank you. Yes. So on week 7, we have a link for the lab module right here. And then for the worksheet, in addition, there's a link for each case study and a link for the dichotomous key. And then there's a link for the infectious disease project because it's due this week, Saturday of this week. But the point is that the way the lab is set up, the instructor sent out all these different case studies. And I thought about putting them all together, but if he changes one, that means I'd have to change everything. And I'd rather just put up his one if he changes one, meaning the lead instructor. So I decided to leave these as separate links. And you have to do all of them or click each of those links. Any question about that? So some learning objectives. You should be able to understand and interpret the results, not so much for the CAMP test, and we'll discuss it, but the bioesculin test you'll be running, the SF broth test, and the optogen test, and be able to use a dichotomous key. You'll need to know how to do a dichotomous key or use one when you're doing your unknown project. And we give you a dichotomous key for the gram-positive infections in the case studies. Realize that this key should only be used for this lab. Do not use it for your unknown project. Oh shoot, this isn't the teacher's study. Just a minute. No, that's the wrong one. I'll get it. Hopefully. Either that or I'll have to wing it. Oh, there we are. I have highlighted when I want to talk, and that's the only difference in the teacher's edition, the teacher's version, I mean. When specimens are received for culturing in a microbiology lab, they're inoculated on culture media that will facilitate the growth of common pathogens from the specimen collected. Like we had last time, the throat pathogens we studied, and normally you grow those on blood agar or chocolate agar. Well, today we're going to be culturing different media because we'll have different specimens. And then you place them in a physical environment to support their growth, and that's usually an incubator. Microbiologists commonly use the colony morphologies and the gram stains to provide important information about the bacteria that's been isolated. However, you can't get down to the species with just those two things, the colony morphology and the gram stain. You further have to do biochemical testing. In this lab, we're going to talk about the CAMP test, the bioesculin test, the SF broth test, and the optogen sensitivity test. CAMP is an acronym for the three microbiologists who invented the test and then described it. It's showing the amplified hemolytic effect that some beta hemolytic organisms have when they're grown physically near Staphylococcus aureus. What you do is you have a plate of blood agar, and then you streak in a single line Staphylococcus aureus down the plate. And then you streak the other bacteria that you want to see if it will have enhanced hemolytic, beta hemolytic activity near the Staphylococcus aureus. You streak that bacteria perpendicular to the Staphylococcus aureus. And then you inoculate it. And I've got a picture of it here. So here's the blood agar, and we streak Staphylococcus aureus down here. Let me see if I can blow this up. And sort of the dark line in the center is the Staphylococcus aureus growth. The clear area is the beta hemolysis that Staphylococcus aureus has. So it lyses the blood cells around the Staphylococcus aureus. And then you streak the other bacteria perpendicular. Streptococcus pyogenes has no enhanced hemolytic activity near Staphylococcus aureus. Listeria has slight enhanced hemolytic activity right here, as opposed to there, near Staphylococcus aureus. Let me shrink that a little bit. And then Streptococcus agaltiae has greatly enhanced hemolytic activity, and that's shown by the arrow, this hemolytic activity here. Let me blow that up a little. Move this over. Staphylococcus aureus, Streptococcus A this way, and then the enhanced hemolytic activity. The enhanced hemolytic activity sometimes looks like an arrow, meaning we have this arrow type shape, and that's the bacteria right there. Any question about that? So that's a positive camp reaction shown here, a slightly positive shown here, and then a negative camp reaction shown here. Any questions about that? The bile esculin hydrolysis test is a tube, it's a slant of bile esculin in the agar. And we can use this test to differentiate between Enterococcus and Streptococcus. Both of these can be beta hemolytic. Some species of both of them are also alpha hemolytic. If the organism hydrolyzes esculin, it will make the product esculatin, which is a dark brown precipitate showing in the tube. So this is negative for hydrolysis of esculin, and this is positive where the esculatin is a dark brown color. You simply need 50% or more of the tube to be darker in color, dark brown or black. And then you say it's positive for esculin hydrolysis. Streptococcus species are negative for bile esculin hydrolysis, and Enterococcus is positive. The SF broth test is looking at Streptococcus faecalis, which has now been changed its name. It used to be called Streptococcus faecalis, but they're now naming it Enterococcus faecalis. But the test is still called SF after the older name. This tube has carbohydrate and sodium azide in it, and then bromocreosol purple. And in the presence of Enterococcus faecalis, it can ferment glucose and make acid products, changing the color of the tube from purple to yellow. So if the tube is purple, it's negative for the SF broth test, meaning no fermentation of glucose. But if it's yellow, it's positive for the SF broth test, meaning it fermented the sugar glucose. Enterococcus faecalis is positive. All of the Streptococcus species are negative for that test. Any questions about that? The optogen sensitivity test is looking at the bacteria and seeing if they're sensitive to this antibiotic. Optogen is an antibiotic. And it is used to differentiate Streptococcus pneumoniae from other alpha hemolytic streptococci. So both Streptococcus pneumoniae and other streptococci are show alpha hemolysis on a blood agar plate. And then we can use this test to differentiate Streptococcus pneumoniae from the other streptococci because Streptococcus pneumoniae is sensitive to optogen, that antibiotic, and the other alpha hemolytic streptococci are not sensitive to optogen. What you simply do is you have your bacteria that's showing alpha hemolytic activity, and that looks like it's dark, and then we plate it on the blood agar and then put down the disc containing the antibiotic optogen and then let it incubate. And if you have a zone of hemolysis, not a zone of hemolysis, sorry, you have a zone of inhibition that's greater than or equal to 14 millimeters, we say that it's sensitive to the antibiotic optogen. And if the zone of inhibition is less than 14 millimeters, and in this case, it's no millimeters, we say it is resistant. You can be resistant if you are less than 14 millimeters, okay? And Streptococcus pneumoniae is sensitive to optogen, so it will have a zone of inhibition, a region showing no growth of bacteria, 14 millimeters or more.
You said it can be resistant even if it's not showing it that way?
Sorry, I'm not sure I understood what you're asking. To be resistant, you simply have to have a zone of inhibition less than 14 millimeters. So if this had been 13 millimeters, that would be resistant, okay? Resistant is not necessarily 0. It's anything from 0 up to 13 or 13.5 millimeters. You have to be 14 millimeters or greater to be sensitive to optogen. Remember, the definition of resistance and sensitivity is you measure the diameter and then compare it to the chart of the standards. And I forget what the abbreviation for the standards are. And the standards are based on how far the antibiotic will diffuse, and then the sensitivity and resistance of the bacteria. Okay? Any question about that? So this is resistant, but if there was a zone of inhibition here, less than 14 millimeters, that would also be resistant. Okay? All right, I think you got that. Anyway, Streptococcus pneumoniae is sensitive. The other streptococci are resistant to this test, the optogen sensitivity test. All right, microbiologists frequently use a dichotomous key that describes which tests have been performed. And in your unknown project, you also need to use this thought process that we call dichotomous logic. What you do is you have a test and there can only be 2 outcomes, and that could be gram positive and gram negative. And then you throw out the gram negatives, and then you start building on that dichotomous key, which builds on the gram positives. Okay, and that is why you're supposed to do 1 test at a time, because if you do more than 1 test at a time, you're not using dichotomous logic. And this is showing you dichotomous logic. Gram stain could be gram positive or gram negative. It's gram positive, so we cross this out, and then we do a test. What test are we doing next? For the gram positive bacillus, we did whether it's an aerobe or an anaerobe. For the gram positive cocci, we do the test whether they're catalase positive or catalase negative. If it was catalase positive, which it was, it's in the family Staphylococcus. And so we throw out the gram positive cocci, which are catalase negative, and that's the Streptococcus or Enterococcus. And then once we have the gram positive cocci, which are catalase positive in the family Staphylococcus, in this key, we then do the coagulase test. If it's coagulase positive, it's Staphylococcus aureus. If it's coagulase negative, it's some other Staphylococcus species, but not Staphylococcus aureus. And then you do the dichotomous key further. Okay? Anyways, that's showing you dichotomous logic. Where the heck is the next to the page? That page is blank for some reason. So we have a video on SF broth and the bile-esculin test. And we also have a video on the CAMP test, so watch those. And you're not physically going to be performing these activities, but you are responsible for knowing how to perform them and interpret them. On the CAMP test, you will be responsible for looking at it and saying, is there enhanced hemolytic activity? Is it slightly enhanced, or is there no enhanced hemolytic activity? And then review how to interpret the following tests and complete this chart. Don't complete them in the lab module. Some of these information you can get from this lab module, but some of them you're going to need to refer to a previous lab module. And like I said, don't do this here. Do that in the worksheet, which is right here. So fill it in on the worksheet. So there's only that above the where you fill in. Fill in this information. And some of it, like I think this was in the last lab. The gram stain, I don't remember when that was. It was several weeks ago, I think. The catalase test, it's not in today's lab. The coagulase isn't in today's lab. The CAMP test is in today's lab. The bile-esculin and SF broth are all in today's lab. I think most of these, oxidase, coagulase, and catalase, were in last week's lab. And these two were probably 2 labs ago. And the gram stain, I don't remember how long ago that was. That may have been 3 labs ago. Okay, just with its positive result, actually the gram stain's done for you. Positive result, it stains purple. Negative result, the gram stain stains pink. So give the interpretation, completing the chart. Fill that in. We have a video on how to use and develop a dichotomous key. It's not exactly part of the lab, but it's explaining things, how to use it. You're not going to use that dichotomous key. You're going to be using the dichotomous key provided for you in Canvas. And these are the bacteria we're going to look at for this lab. And then do the case studies. Do case study 1, you'll read about it. Let's go ahead and open that. There it is. Come on. So read about it and take a look at the tests that were done initially. This is the wound on the boy, or whatever you want to call it. And this looks like a gram stain. Is that what that is? Shoot. I think that's the gram stain. I'm not seeing it here. There it is, gram stain. And then there they cultured it on a blood agar plate, and that looks like alpha hemolysis. You're supposed to interpret that. And then they did, taking from this bacteria, put it on this, and then did the hydrogen peroxide test. And there's the result from the hydrogen peroxide test, meaning the catalase test, whether it's catalase positive or negative. Come on. And then they took this growth and then grew it on a mannitol salt agar plate, streaked it out, and the mannitol salt agar looked here. If it remains purple, it's not fermenting mannitol, the sugar mannitol. If it can grow on the mannitol salt agar plate, that means it can tolerate 7% sodium chloride. So this has salt in it, 7%. And then if it turns yellow, it ferments the sugar mannitol. So those are the different tests you have for case study 1. And then give me the name of the bacteria that was isolated. And like I said, if you want a little bit of help here, in the lab module, it tells you the bacteria we are considering. Where do I have that? Here it is. These are the bacteria you need to consider, and take a look at the dichotomous keys. It'll help you identify the species. Any question about case study 1? Do that the same, write in your answer then for what you believe the species to be right here, although you do it on the worksheet. So right here. And then take a look at case study 2 and state which species is giving the infection, and do that for all of the case studies. There's 8 case studies here. And as you know, the case studies do take a little bit more time. So expect this to take a little more time, because it'll probably take you 5 to 10 minutes for each case study. All right, any questions? If not, I will be here to answer questions until the last student leaves. There is one student I need to talk to about the unknown project. And if she wants to do that while other students are here, we will do that discussion here. Otherwise, we'll have to wait until the end of the lab to have that discussion. Is that student here?
Yes, I'm here.
Okay. Sorry, I didn't see who's talking, so.
No, you're totally fine. I can wait
Okay.
until after everybody. That is a-okay. That's what I planned on doing anyways.
Okay, all right. Mary, all right. Okay, so I will be here until the last student logs out, except for Mary, who will not log out. And then we will have our discussion. All right, so get started on the lab. There's only one other student, so when that student logs off, Mary, we can talk.
Actually, it's just us.
Oh, good, all right, then we can talk. If you want to private, keep an eye on the number of participants. We usually hear a bell or a ring. If somebody-
Yeah. No, I appreciate that. Thank you so much. So I think a lot of what you said kind of cleared up a little bit of it for me, I guess a little bit of my panic. Instead of panicking about the unknown, I need to panic about the infectious disease report. So that's good to know. I need to switch my focus. But I still am very behind on the unknown lab because I was not submitting my request correctly in the right form.
Okay, you're behind, but you're not very far behind.
Okay. Well, that's good. Now that so in my head originally, I thought it was flip-flopped. I thought the infectious disease report was due later in the quarter, like December, like when December 3rd. and that the unknown lab was due this weekend. So I had them flip-flopped in my head. That's why I thought I was so incredibly behind on the unknown lab.
Uh-huh.
So
Okay.
now that I understand I'm not that incredibly behind on the lab, I think I'm going to kind of table that for a minute. And I was looking through the infectious disease report, and it seems all pretty just straightforward. Like I looked through the outline, obviously, that you have for all the what we have to have in it and what have you. And it looks pretty upfront and what have you. I just need to get it done, obviously. So I actually don't think I need any clarification any further.
Okay.
The only thing I did see, since I have your
Hold,
attention.
hold it, Mary. Hold it. It just occurred to me.